Yes. So how loaded is that? It's a really short title. It says the hope and reality of medical marijuana. It has asterisks, footnotes, and air quotations around it. So you know that it's not going to go exactly the way we thought it was going to go. I was hoping I'd be able to come out and show you all kinds of forest plots to show you that, oh, the evidence is just absolutely fantastic. This is the best, you know, oh, everyone should be on this. We should, you know, just vaping ourselves this week, you know. Unfortunately, that may not be the case, but there has been a lot of change in the last little while. Just to be clear, yes, I am a consultant for Aurinia Pharmaceuticals. I do pharmaco PKPD type stuff, lupus nephritis. But the only reason I'm a consultant is because every time I work for any kind of commercial organization, they go out of business. So it's going to be interesting to see how long Aurinia lasts, okay? The other thing is, most definitely, medicinal uses of marijuana are off-label, okay? Except for a few things that they actually have. And I don't think there's any money. No one gave me any money. Steve brought cookies that I should have brought brownies, but they would have been suspect. Yeah, we could have thrown Doritos out like Trump throwing, you know, paper towels. The other thing is, if you get the munchies halfway through this, it is no way that can be a causal relationship. It just happens to be this time of day, okay? So really, what I really want to do is I want to provide you the background. There is a lot of new information that's come out. It's not definitely at the cardiology level of evidence that Margaret was kind of you were disparaging. You were dissing a little bit, yeah. But what I really want to do is, this is going to be legalized. This is where we're at. It seems like a year and a half ago I was talking about MAID, and it was the same sort of thing. It was decriminalized, but we didn't have legislation yet. But this is kind of the void we're in right now as well. And your patients are going to be asking for it. So when they asked me to give this talk, I said, I'd really like to, and thank you for the opportunity, because I have patients now. It used to be younger patients. Now I'm getting 90-year-old grannies going, could I try the medical marijuana, right? And it's like, and we need to be able to give them the best information that we possibly can. The other thing is, as I say, is it hope or is it hype? I think for the first time possibly in my career, it is more hope than possibly hype, but there is a lot of hype. So coming along with the perfect storm that Steve was talking about, the opioid crisis, is there's a lot of conjecture that pot might be able to replace it. And people, of course, don't die from pot, or do they? He says, leading. Anyhow, this came out in Science, which is a pretty reputable journal. This is not a scientific publication. This is his opinion. And it's the most bizarre thing I've ever read, because he goes into the opioid crisis and he talks about all the people that are dying of opioid overdose, and then he hardly gives any kind of information whatsoever on, does marijuana actually work for pain? And actually, if you look, we're going to go into some data, but if you look at nociceptive pain, which opioids do work pretty well for, we don't really see that coming out of just medicinal marijuana or marijuana used for medicinal purposes. What do we do, though, when we see this sort of thing? So here's a young boy who has drug-resistant seizures. And he actually, it turns out he has a variant of epilepsy. He was having up to 100 seizures a day. Nothing would stop it. So they gave him cannabis oil, whatever the heck that is. And then for the last 300 days, he hasn't had it. This is, and you hear this coming out of Colorado. People were moving down to Colorado so their children could get cannabidiol oil in order to treat this type of seizures. This actually came out, what was it? It was September 6th, 2017, and he was the first one in the UK to actually receive a prescription for medical marijuana. What do we do for those patients that we have this unmet clinical need? Well, we continually look for better and novel treatments, because we want to be the advocate for our patients. When patients ask me things, I try to be open-minded. I try to come up with the very best information that I can. And I sure wish I had a cerebral enhancitron, because I really think that that's what we always need to do as pharmacists, is look for a way to find answers. Now this actually was, and again, you just can't read something without reading about medicinal uses of marijuana. And this came out in Alternet, and this was glaucoma and medical marijuana. Yeah. Yeah, Alternet and an alternate reality. And it was written by a guy called Trey Reckling, who also writes for the Fresh Toast. Anyhow, he's talking about, there is some selection bias here, right? So, but anyhow, he says, oh no, I know this stuff is great, but the catch would be is tetrahydrocannabinol has a short half-life, so you have to smoke up all day long. You're stoned 24/7. Not so, he says. THC's depressurizing effect is not dose dependent. Well, that made me really nervous as a pharmacokineticist because he's mixing his dynamics and his kinetics. Dose dependency, oh my lord, it's nonlinear, right? No, so what he's talking about is it doesn't seem like you need a very high concentration of THC to actually decrease your intraocular pressure. But how many people have had patients taking marijuana for glaucoma? Well, I have. I must have tracked them. And, one thing that's really interesting is what the guy said to me. He said, I got some of that government-grown stuff, in the Defen bunker. And he says, I hardly got a buzz off it at all. And I said, well, how's your intraocular pressure? Oh, it's fine. So the thing that was interesting about this, I'm just going to, if I see the mouse up there, is he's the founder of the Academy of Cannabis Science. That sounded pretty good to me. I'm looking for information, right? So I went to the Academy of Cannabis Science, and wow, medical marijuana consultant certificate program. It's got George Washington and the Greater Seal of Washington State. George Washington would not lie to me. It's got pain management and cannabis therapy and beyond folklore and pseudoscience. It sounds pretty good, okay? But when you go and you dig a little deeper, it's the most bizarre website I've ever been on. So in recent University of California study, 92% of patients preferred cannabis to opioids. That sounds pretty good. No error bars, no p-values, nothing. But I was interested. But this is all bizarre. Well, Washington was a patriot, a president, and a hemp farmer. And there's Richard Pryor, of all people, talking about the Constitution. So it appears that not believing in cannabis science is both unconstitutional and unpatriotic. So, but I clicked on this. I was hoping this would take me to a link to a study. No, the box just got bigger. So, okay, now I've kind of set up a straw man. I'm setting him down. If you really want to better, go look at poster number 10. Dr. Nesha Yuxel and her, they did a wonderful assessment of the marijuana information you get off websites. And yes, it tends to be unidirectional. It's all great. There's no downside. It's just a nutraceutical. It's the herb, right? And that just isn't how drugs work, unfortunately. So how are we doing in Canada? Well, first thing, you go to the source of all information, the government website. And if you look at the Justice Laws website, what you find is that the marijuana for medicinal purposes regulations has been repealed. And then if you go and dig a little bit deeper, you'll find, oh, there's really no regulations right now because they have been consolidated. So what we're waiting for is we're waiting for legislation. And we're not sure exactly what that legislation looks like. Does that mean you can't get marijuana for medicinal purposes? No, you can get it. It's actually quite easy to get it. Click, 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 and you've got it. So what we say in Canada is, well, there are more guidelines than actual rules, right? So we're waiting for rules. We're not exactly sure what that's going to look like. But after the repeal, then the Canadian Research Institute for Substance Abuse and the Centre for Addiction and Mental Health came out with Canada's lower risk cannabis use guidelines. And they published them in the American Journal of Public Health. And what's really interesting is their first recommendation is if you don't want to risk anything, don't take it. It makes a lot of sense. That does us no good as pharmacists because if somebody has an unmet clinical need and they could use drug therapy, that's a drug-related problem, right? So the second recommendation, which was interesting, is they are talking about something called THC and CBD levels and ratios. And so we do need to talk about that in a little more detail. Now, of course, benefit risk is, I worked in the drug industry for, 15 years or something. I don't know. How old am I? I've lost count. But the fact is benefit risk for recreational use is way different than for medicinal use. So for recreational use, it's a subjective kind of thing. The benefit of the recreation seems to outweigh the risk, and it's often based on a previous lack of harm experience. Of course, we launched the shuttle at this temperature before and never had any problem before. So that kind of previous, that anecdotal previous, I didn't get hurt before, that's no justification. That's no guarantee you're not going to get into trouble. This is almost quaint now, because where I'm going to go with this is what's happened to marijuana down through the years. So there's good old Cheech and Chong, and they've rolled a pretty big doobie there. That's the only way they're going to get the kind of doses that people would get out of one cigarette today. Now, the other thing is if we're talking for medicinal use, it's disease acuity. As soon as your disease state changes, especially if you have a life-threatening illness, you're going to put up with a lot more risk. So, and it's also with poor quality of life. So for instance, a good example is chemotherapy. You'll put up with highly toxic chemotherapy because you're trying to save someone's life. And anybody who's been through chemotherapy knows it's whack-a-mole, right? I'm feeling much better today. They say, that's really good. And then they give you the next dose, right? You're not going to put up with that, though, on a recreational basis, the risk benefit's not there. Also for accepting higher for quality of life. And of course, we get that in palliative care all the time, the so-called principle of double effect. What we're putting people on drugs for is to improve their quality of life. If it might have the unintended effect of shortening their life, that was not our intention. Seems like a... The popular websites also do this. Lots of numbers. And I'm not arguing with anybody that tobacco probably does cause that. I don't know where this data is from. Like, I don't know what year it's from. I don't know what country it's from. And if I have a peanut allergy, it's possible that marijuana might be safer than peanuts. But I honestly don't know. And this is the kind of thing that I do find very frustrating. Because especially in clinical science and clinical research, we have standards that allow us to make sort of decisions, informed decisions. So that leads me to what happens when a drug, now I wanted to just share this very quickly. The European Medicines Agency had a 5-year program, and they worked very hard to decide on what kind of information do we need to assess benefit risk. And they came just shy of saying you always must have quantitative data to prove risk benefit. So what they actually do is, no, they did say you can allow qualitative, you'll have an effects table. Every study that's ever done is on the effect. So you look at favorable effects and you look at unfavorable effects. So in one table, you should be able to assess the drug you're looking at, know how well it worked, but what are the unwanted effects? What are the safety catches? The other thing that they put forward was multiple criteria decision analysis. Love to teach a course on that, by the way, just saying. It would really be cool. The other thing is at health technology assessment, we expect there to be some sort of systematic literature review, expert consultation, targeted examination. It says methodological, even going into the gray literature. I do want to warn you, I've spent a lot of time in the gray literature. This is what happens to you. It rubs off, okay? The other thing is consultation with advanced partners, and then a wider dissemination of consultation activities. And we expect to see some sort of diagram like this where we can see everything that they've looked at. Now this is what's interesting too, are these value dimensions. So you're looking at the burden of disease, therapeutic, was the endpoint, was it a direct, was it surrogate? We also want safety. Now innovation is clinical novelty, but that's also the science, the preclinical science. The preclinical science promises all sorts of things, but when you get into people, sometimes it can be very, very different. And they also look at socioeconomic factors. You cannot do a quality of life assessment, a proper pharmacoeconomic assessment of a drug without looking at socioeconomic factors. They then pull it all together in what's called an advanced value tree. Now here's this stuff down here, the mechanism of action. This is innovation level stuff. This is where an awful lot of the marijuana literature is right now. But that sometimes gets taken out into the popular milieu as, this scientists have proven, ... And we see that all the time. So that's the level of scrutiny that a drug really has to undergo before we can actually say that the benefit risk is favorable. So why do people accept all sorts of different things for instance, medical marijuana? So now we have my philosophy prof is coming in, rudely interrupting epistemology. Epistemology is that branch of philosophy that deals with the truth, okay? So now I've gone about it, you start with the Greeks and you go forward. This is probably about in the Middle Ages right here. Knowledge versus belief and reliabilism. We tend to say it this way. If I know something, I don't have to believe in it anymore. But if I have a very, very strongly held belief, that doesn't necessarily constitute knowledge. So one of the things that reliabilism argues is that S knows this thing P, and the IFF means if and only if P is true, okay? But S believes in this thing P, and S belief is produced by some sort of reliable cognitive process. So how does that work? How do I know that the Earth is round? Okay, astronauts know that the Earth is round because they've been orbiting around the planet. They're pretty sure about it. So they can actually say it's no longer a belief of mine, it's knowledge. I've not orbited around the Earth, but I can actually talk to them. I also can study physics. I also can get on an airplane and fly and go through the terminus from light to dark and dark to light, back again, and things like that. So I can come up with a pretty strongly held belief that approaches knowledge on something like this. The reason why I bring this up is this was just published this last month, and it was epistemic beliefs' role in promoting misperception and conspiracist ideation. And I bring this up because here we are talking about the legalization of marijuana. What kind of science would we like to see as clinicians before we can recommend it for our patients? And when you have people that go online and they're absolutely convinced that this is the best drug that there is out there. And what was interesting about this paper, and it's worth, it came out in PLOS One, is, and they surveyed, and it was in the United States, but I suspect it would be the same in Canada. About 48% of the people surveyed said, it's my gut. My bottom line is the gut check. I feel that this has got to be true. Whereas down here, it's like, no, we have a need for evidence. Evidence is more important than whether something feels true. I trust the facts, not my instincts. Tell me what is true. And a hunch needs to be confirmed with data. That's kind of where that's where we're trained. That's where we're coming from. The scary part that I've come up against, at least lately, is that scientific conclusions can be shaped by politics. So that all of a sudden you can come out and say, no, you have a hidden agenda. Your drugs, I don't trust your drugs because I know you're just trying to control me or something like that. But this guy tells me it's not the same way. So I brought that up because this is where we're at in the whole marijuana discussion. We in clinical pharmacy, we have a reliable cognitive process, and it's called evidence-based medicine, okay? And so my whole problem, though, with a lot of the medical marijuana literature is, yeah, we're actually seeing some RCTs now. They're tiny, they're small sample size, and the biggest problem that all the literature, all the epidemiology, all the observational trials is what the heck was in that plant, okay? Where's the standardization that we can actually look and say that we know exactly what they got? Now, in my mind, legalizing this will have one effect, and I have a bunch of friends in health policy that agree with me, is that at the very least, we'll be able to put some standards on it. And you know how much THC there's in it and how much CBD, and maybe we can then make our epidemiology will get better because we have a better estimate of exposure, okay? But that, to me, is how that works. Now, other people have different reliable cognitive process. So what I've tried to do is I'm going to put in juxtaposition wherever I can what you find on some of the websites and what some of the papers say. Now, the other thing is cannabis is classified into 3 species, sativa, indica, and ruderalis. Well, no, botany doesn't agree with that. Actually, ruderalis may be part of sativa. The thing that's interesting about this, though, is they are starting to talk, if you can make that out down here, is, can you see that? Yeah. So sativa tends to put out more THC than CBD, okay? And then, but they got it all worked out very nicely. This is the website if you want to go on it. And it actually takes you through this little tutorial before you get to the, and are you ready to buy window? Yep. So, wouldn't you love it if drugs were that way? And during the day, you should take sativa, and at night, you should take indica. And they've got it all worked out, which seems quite nice. And they have this lovely little, so in all fairness, I look at sort of populist RCP, rational cognitive process, and it's this weird mix of pseudoscience. There's some basic pharmacology and chemistry thrown in. You'll even see sometimes they actually do quote a paper, but it's usually like from Annals of Chemistry or something. So it's not anywhere near the level we need in clinical land, but there is biological plausibility, okay? There's some existing epidemiology, tons of anecdotes. Well, look at me, I've smoked marijuana for years and I'm only 26 years old, you know? Anecdotes, testimonials, there's certainly trial and error, and there is marketing. There's a lot of marketing. So I just wanted you just to share that with you so you can see it. This is actually a summary that came out in 2006 in TIPS, Trends in Pharmacological Sciences, is always that you have these big, massive review papers. And what's interesting is that, well, we've actually had marijuana around. So the Chinese in their pharmacopoeia, they had actually categorized it in about 200 common era. And then you get, has anybody ever heard of Sir W.B. O'Shaughnessy before? He apparently did a lot of work methodically, looking at the medical properties of cannabis. I haven't read too much about him, so I'm sort of wondering if he did the experiments on himself or not. What's interesting to me is I grew up through the 70s and everything is, yeah, they say the cannabinoid pharmacology is thoroughly investigated through the 1970s to the 1990s. But the receptors themselves weren't cloned until the 1990s, okay? So there was a lot of things that weren't known. The other thing is the first cannabinoid receptor was found accidentally. They were studying what they thought was an opioid receptor, sigma receptor, which was considered to be promiscuous because it binded with other compounds. That's inappropriate. But then they actually started synthesizing. Once the question then became, just like with opioids, is if opium poppy has all these effects on you, you must have endogenous opioids. The same question was asked here. If you have this plant that seems to elicit all these responses, there must be endogenous cannabinoids. And they found them. And there's actually 2 key ones, and we'll talk about those. The other thing is you move forward into the 2000s, we actually see approval of, and again, Sativex certainly is a plant product that's been standardized, and they actually went through all the trouble to get it on the market, and good for them. So we actually can assess a risk benefit, but they're also making all kinds of synthetic compounds as well. So if we look at why are cannabinoids implicated in so many disease states, well, a lot of it is because of biological plausibility. A lot of this is this innovative step. And if you go and you look at it, so we have basically, you have 2-AG and you have AEA, and of course, it's anandamide, sorry, is from the Sanskrit and Pali words anida, which means joy, bliss, and delight, and amide. I don't know that for a fact. I believe it because it came from so many different sources that I think it probably is right. But I got to admit, my Sanskrit and Pali is not what it used to be. Okay, so the effects are mediated by 2 receptors, and they tend to be CB1 in the nervous system and in the periphery. It's never that easy, right? So if you actually go and look at these, well, the problem is, first of all, is AEA will actually bind to CB1, but it actually can bind to CB2, and 2-AG can bind to either one of them, okay? If you go and you look, first and foremost, they're coupled to G inhibitory and G other proteins. What do you expect from G protein coupled drugs? And can you think of another class of drugs that you know about, that might have heard about this weekend that's G protein coupled? Even G inhibitory protein. Someone said opioids. And read lips. Okay, yeah, absolutely. And so what do you expect though from G? So one of the things about G protein coupled drugs is that amplification of the signal across the membrane can be like 10 to the 6th, 10 to the 9th. Very, very potent, can have very profound effects. But the body has to deal with that and says, you know, if you were on this stuff all the time, I can't handle that. So what do you expect from G protein coupled drugs? Tolerance, absolutely. And that's going to be a big problem when they go to actually look and see if they want a saliva test for marijuana. It's like, but if you've been taking this stuff and smoking up constantly, you're going to have very, very high levels, but you may not be actually intoxicated because you've developed so much tolerance. The other thing about these is THC is a partial agonist at both. You can tell by their alternate names that CB1 does tend to be in the central nervous system. But if you go down and you look further on, it does show up in some in leukocytes in the testes. Actually been implicated in testicular cancer. The other thing is that if it's CB2 that tends to have, believed should have the more immunological effects. So if you go and you look, and we love diagrams like this, and I can just spend all my day marveling at them, we see that we have CB1 or CB2, and certainly cyclic AMP, but when we start looking at some of these, you know, these effector molecules, you start to say, oh my gosh, depending on where this is, these could have huge effects. Nitric oxide. So if we have nitric oxide, we could have vasodilatory properties, could have effect on platelets. We could have immunomodulatory activity. If you actually go over here and look at GPR55, which is another G protein coupled, it affects platelet adhesion, it affects hypotension. So we should have cardiovascular effects from this drug. If you go intracellularly, you see that it affects gene transcription. It affects apoptosis. So maybe it has some effects in cancer. Now that's perfectly reasonable in the innovative part of the equation to say, now we want to go look for that and see if we can come up with drugs that are useful. So this actually came from TIPS. So you can see, we're not going to go through all this. But you can see why they're doing studies on virtually everything. I've never seen a single group compound that is being looked at for so many different disease states. But remember, a lot of these studies were done in knockout mice. You know they're knockouts because they're wearing evening gowns. This is still, the problem with knockout, a lot of knockout trials is that you knock out a gene and you expect it to do one specific thing, only you found out it actually did way more than that. So some of this early stuff is in that sort of level. If you go and you look at pain, everywhere all the places that you expect to see modulation of pain signals, there's a receptor. There's a cannabinoid receptor there as well. And we can go on, but if you go to the popular website, everything's much clearer there. Oh, we have the little lock and the key, and the THC hits the one and it doesn't hit the other, which we know isn't true. But anyhow, the one thing I gotta say for them, they're really good at their messaging. And maybe I keep thinking, maybe I should be a little better in my messaging to my patients. The other thing is, if you're really looking at a very nice, this just came out in 2017, I think September. It's the cannabinoid signaling in health and disease. It's in the Canadian Journal of Physiology and Pharmacology, and it looks at everything that they're studying. And it's really thorough, it's really well done, but a lot of these are models, they're animal models, so it's a good review. He's cute, right? So biological plausibility becomes a very strong rational cognitive process that is probably being taken out into the popular literature as well. Cannabinoids in cancer, and this came out in 2008. Well, it's quite compelling because you go and you look at the different types of cancer, and you see different expression of cannabinoids, and you see different type expression of the receptors. So it's hard to believe that there wouldn't be a biological mechanism there. And of course there is. And again, what it really boils down to is it affects tumor progression, metastases, angiogenesis, but it also causes, it affects apoptosis. So again, we think there's a lot of smoke here, but we don't, but what we really need are some bona fide clinical trials. The other thing is I've never seen, again, well, maybe it works in sepsis, so you're going to be, well, you're going to be in good shape. It might be for cholera. And this one was really good, an FAH far-fetched approach to treat osteo. That's a hilarious joke once we get into the F fatty acid acyl amide hydroxyl hydrolase, right? So but this is what a lot of the clinical studies we have look like. And this one actually just came out March 2017. Low-dose cannabidiol is safe but not effective for Crohn's disease. It's about 20 patients. It's a small proof of concept. They did standardize. It's 10 milligrams of CBD BID, okay? And then they did their endpoint was actually the Crohn's disease activity index, okay? It's in the literature, so they're trying. So this is really a proof of concept type study. They found at the end of 8 weeks it really didn't have much effect. And it was registered with the FDA and everything, but we need to see more of it like this. I'm encouraging this, not just criticizing it for being too small and all sorts of things. But it is good that they're starting to do that. Unfortunately, this is the kind of stuff that if it was positive, gets lifted out and say, scientists today have proven, ... There's a whole bunch of things about pain, cannabis species, and oh, wait a minute, there's a use disorder. Cannabis use disorder. Oh no, Cheech and Chong said the good thing about this was that you never got addicted. There was no withdrawal. No, that's not true anymore. And there are people that actually say, I cannot stop smoking cannabis. So we're left with this. It could work. And I just love Gene Wilder. And that was Young Frankenstein. That was after he spent all night figuring out if he could make a monster. So you go to the Cochrane reviews, and they're ongoing. You go and you look, and they're almost updating these things daily because new papers are coming out. But if you go and you look at the Cochrane reviews, what they're going to say, and you're going to see a lovely forest plot, which I love to see, favors not the treatment, or that the variability is so humongously wide, we just can't do it. The other thing is, which is based on chi-squared, it's Greek letter iota, right? I give an iota because the heterogeneity in most of these studies is horrible. So the other thing is, this came out in 2017 in British Medical Journal. The reviews failed to advise on cannabis use because of poor research. That might be a bit hard. I was actually thinking it's poor quality research because there's actually a lot of research out there. The problem isn't a lack of studies, it's a lack of high quality studies. And it's a lack of reproducible standards of marijuana. And of course, this is Thomas Henry Huxley who said, the slaying of a beautiful hypothesis by an ugly fact. And you got to listen to someone with sideburns like that. I had sideburns through the 70s, but they weren't that big. So this just came out, and I thought, I was working this, and I actually worked up a whole bunch of different papers, but I thought, no, this is a good opportunity. How many people are members of the National Academy of Science? You can join for free, okay? And the other thing is the National Academies of Press, their government may be anti-science, but they've got some good scientists down there. And this, all their books, so this just came out in just 2 months ago. The health effects of cannabis and cannabinoids, the current state of evidence and recommendations for research. And so I thought I'd share this with you. Now it's 486 pages and they will charge you for that. But if you download it as a PDF, it's completely free. So I don't know, so I've signed up for this. So every month I get the National Academies of Press has come out with this. There's leadership stuff, there's stuff on infectious disease, there's all kinds of things. So I'm downloading stuff all the time, my hard drive's filling up, and my wife is like, what are you doing in there? Are you reading more National Academies of Press stuff? Yeah, because it's so good. So, I think I said something I shouldn't. Anyhow, the point is they did a very, very thorough review because there had been so much new information that had come out. Now, it's not at the level of a Cochrane review, and they make that very clear that they weren't tasked to conduct multiple systematic reviews, but they did adopt basically the same process. They did a literature search, they had assessments by more than one person, they were looking for bias, they went through key literature, and they did make not grade level assessments, but grade-like assessments. I call it downgrading. So I don't know how clear this is, but you can see, so they did, this is their process, literature search, they did a systematic review, they went to the primary literature, and then they did data synthesis, and then they weighted the evidence. And you can see that they have all their searches, and all their search terms show up in the book. And the fact is their last searches were in December 2016. And you can see they looked at injury and death, therapeutic, all different therapeutic categories, cancer and immunity, psychosocial, mental health, pregnancy, and cardiovascular respiratory. They're basically trying to pull together information that you kind of need if you were going to file for an NDA that they don't have. So they're trying to see if they can pull enough stuff together to know how bad is this really going to be. Now, it's interesting too that in this, they also look at all the states that have already legalized and what are the prescriptions going out for medicinal marijuana. Well, severe pains, number one. Muscle spasms, pretty well due to MS, number two. Nausea, pretty well related to chemotherapy. I can tell you right now, that's where there's the most evidence, you know? So, and then they get down into seizures and epilepsy, cachexia, glaucoma, HIV, AIDS, and so forth. So they went through, and we don't have time to go through this in great detail, but they went through all, and they really looked thoroughly. So it's worth downloading, okay? And they looked at injury and death, they looked at psychosocial, they looked at, and again, normally we would never put a drug into a human being until you'd at least done a repo tox study in animals, right? So now you've got people pregnant and smoking because it's completely safe. They also, this was their evidence categories: conclusive evidence, substantial evidence, moderate evidence, limited evidence, and they have standards in each one of those. No or insufficient evidence to support the association. So I can tell you, as far as evidence-based medicine goes, it's pain, antiemetic, and MS spasticity. There was some moderate evidence for sleep disturbances secondary to other disease states. Now remember, we're not quite at the Cochrane level, but this is where it's at. The other thing, there was no evidence to support or refute that it actually is working in cancer, and this is despite there's been a huge amount of research in glioma. And we don't have good drugs for glioma. But despite that, they actually couldn't conclude that we know for sure that it's working well in glioma. They also said it's not good for irritable bowel syndrome. Have you ever heard of irritable spouse syndrome? That's what my wife suffered from. And epilepsy, they said there's nothing with epilepsy, but I had to share with you this because it just came out, what, 2 months ago? So cannabidiols for epilepsy, and the editorial was real data at last. And I don't know if I'm going to take the red pill or the blue pill on this one quite yet. But the fact is that in Dravet syndrome, which is a loss of function mutation at the SCN1A gene, they took 120 children. They looked at their primary endpoint was convulsive seizure frequency at 14 weeks compared to a 4-week baseline. They had a standardized cannabidiol oral solution that was 100 milligrams per mil that they titrated up to 20 milligrams per kilogram BID. This is the company that funded it, and this is the company that's actually trying to file based on some of this data. I wanted to go through it very quickly, but you did see how many people were screened, who got randomized, and what happened to them. There's a pretty high withdrawal rate on the CBD group. The other thing was, these were children 9 years of old, was their mean and median, but it was typical of this syndrome. They tried on average almost 5 drugs. They were currently on 3 drugs. They'd been on every drug that hadn't worked. They'd even been on ketogenic diets and vagus nerve stimulation. And what they found, and this just blew my mind. So here's the cannabidiol group, 12.4, this is the median, convulsive seizures per month. Look at the range, 3.9 to 1,717. Yeah, they did non-parametric tests because this is not a normally distributed sample by any stretch of the imagination. The other thing is in the treatment period, it definitely comes down, and they hit their endpoint using Wilcoxon rank sum and Hodges-Lehmann estimator, and we could argue about that if we want, but let's not do it right now. But even if you look at the decrease, it was 0 to 2,159 seizures. So you got to realize this is definitely an unmet clinical need. This is something that out of compassion, I can completely see why they've done this study. And they have done a randomized controlled trial. They found out that it works pretty well for tonic-clonic seizures and possibly partial, but not for anything else. The other thing is, unlike the websites, oh, lo and behold, if it does work, there are also adverse events. And the fact is diarrhea was one of the big ones, with somnolence was the other. So then they concluded, which is usually the way you always conclude these kinds of studies, additional data are needed to determine the long-term efficacy. But there probably will be a product. And I think under these conditions, it probably makes sense under an orphan drug type condition, right? The risk for cancer, and again, how are we doing? I don't even know what time it is. What day is it? Who am I? Okay. This is good. Okay. So they did look at, and this was the risk of lung cancer if you were smoking. And there was only moderate evidence, but there seemed no association between lung and head and neck cancers, which is nice to know, but I wonder about it. Limited evidence for MI and subarachnoid hemorrhage. Also, there's been all kinds of claims that it'll stop diabetes, it's great for diabetes. Well, they couldn't find anything that. Certainly, if you have respiratory problems, smoking marijuana is not going to help you. The other thing was substantial evidence for car crashes. Gee, I wonder why. Also, there was substantial evidence for low birth weights. Although some of the papers, the mothers were smoking cigarettes as well, right? So, and that certainly can cause low birth rate. Or sorry, birth weights, yeah. Moderate evidence, there was moderate evidence for impaired cognition, learning, or memory, or attention, which is Wade from high school. Amen. So the other thing was not all the psychological effects were harms. There was some evidence for better cognitive performance in some psychotic disorders. But if you're at risk for developing schizophrenia, smoking marijuana is not going to help you. So now we want to use medical marijuana. So my point here is that marijuana certainly has not been evaluated. And we're talking plant product marijuana, not synthetic things that have gone through, or even Sativex that has actually gone through and been approved for specific uses. We're talking about marijuana that people are talking about buying and using for medicinal purposes. It has not been through any kind of standard process that we would approve medicine. Can it be standardized as a plant species for medicinal use? Sure it can. And there's even a guidance for that. Oh, there's an app for that. There's a guidance for practically everything. And there is a guidance for developing plant products and standardizing them. Can the plant be modified by breeding or genomic alterations? Going on in Vancouver right now, okay? Should medicinal compounds should we only use synthetic compounds? That's a possibility. But in the meanwhile, for these unmet clinical needs, when you have a patient where you're really struggling, what should we do? So I wanted to share with you a couple of cases of patients that I've been involved with. And this lady hit us particularly hard. She was wonderful. She was only 48 years old, and I know for some of you go, 48, that's old, but for me, it's looking younger all the time. She was a writer. She was married with 2 sons, and actually her children were quite young. She had a T4N1M1 squamous cell carcinoma. It was of the left tongue. It was diagnosed in 2007. She had it resected with a flap. She had radiation therapy. She had a ton of radiation. The number of cycles was mind-boggling, and it was completed in 2008. She had horrible side effects because of the RT. She had really bad neuropathic pain as a consequence of it. Stomatitis, she couldn't eat, she couldn't swallow. That's the thing about head and neck cancers. There's just no place for a tumor to grow, right? So she did pretty well until 2014, and then it recurred. It was hypopharyngeal. She ended up with a total laryngectomy, pharyngectomy, total thyroidectomy, and the tumor was invading the right common carotid artery, and so she'd bleed. And it happened. And this was quite literally, this is what happens in my world, is so cancer is a prothrombotic event or a disease, right? So you should have them on VTE prophylaxis. At the same time, she's bleeding all the time. So they basically crazy glued it because it was actually exposed. And the fact is, then we had this discussion, well, should we put her back on VTE prophylaxis? So we actually will talk to patients and say, how would you prefer to die? With a clot or bleeding out? And we actually ask people, not that bluntly, but that's really what happens. Her pain was in the right lower neck, right posterior neck, went down into her shoulder. It was somatic nociceptive with definite neuropathic features. And she had lancinating pain. The word lancinating should be a boop, 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 boop. That sounds neuropathic. Right hand numbness with a C7 distribution, which was aggravated by writing, and that's the only way she could communicate with us anymore. And you'd go in to meet her. She was just wonderful. She had beautiful handwriting. She had beautiful prose. She had tablets of paper. And you'd go in there, you'd be in there an hour at least, just to ask them, and she would write all these things. And her prose was just beautiful because she worked as a writer. If I had prose like that, I probably wouldn't be here. I'd be writing novels or something. The other thing is the pain kept her awake at night. So she had episodes, she was awakened most by pain through the night. So she also had depression and anxiety on the ESAS scores. She had multiple awakenings due to pain. Yeah, she was also anemic and things like that. That wasn't really the biggest problem that we were having to deal with. She had hypothyroidism, GERD, and C-spine osteoarthritis. And according to everything that I read, she had no known drug allergies. So I actually went in, one of the things I do is a very, very thorough, what we call a functional BPMH, looking for phenotypes is what we're doing. And lo and behold, she described almost to the letter a beautiful serotonergic syndrome with venlafaxine, even though there was I could find no record whatsoever she'd ever been on venlafaxine, but it was a physician samples. Got to enter those. So the other thing, we're not dispense them at all. We're pretty thorough in our analgesic histories because we're trying to figure out where they are, what's worked, what hasn't worked. You can see I did approximate MADDs. The problem was that she didn't really like taking any of these drugs. So she was on Percocet. She'd take them about 5 a day. She got around 45 morphine equivalent daily dose. She got switched to hydromorphone. Again, she was at a lower dose, but it didn't really work very well. Her Tylenol 3, she pushed all the way up, so she was getting codeine at 240 milligrams a day, which is around, so you can see that the MADDs are starting to kind of add up. And she was on morphine when she came in, but still didn't have great efficacy. She'd been put on gabapentin, but by the time we see her, she's having a terrible time swallowing capsules. So we're not sure how much she's taking. The mirtazapine went from 15 to 30. She still wasn't sleeping, but we also weren't sure how much she was taking. And she had Tramacet lurking about and coming in and out of there. And it's an SNRI, right? So the fact is we weren't really sure what was going on. So doc sends me up and says, do your little thing, Pat. So I go up and I'm looking. And so what I'm looking for is what was the response to the drug? And we tried to come up with kind of I tried all kinds of different scores, but the doc said they were too complicated. So I said, if you're up there talking to them and you're not getting the effect you want, just give them a -1. If you're getting more than the effect you wanted, give them a +1. And if everyone said, it seems to work, it's okay, just give them a 0. So then if you get a whole bunch of -1s or you get a whole bunch of plus, it adds up. I can actually go up and then I start looking for, is there some sort of common pattern? So that's what we're trying to do is use their med history and the response that they have to try to come up with a possible of a phenotype. Well, Percocet goes through 2D6 for its conversion from oxycodone to oxymorphone. If you're an ultra metabolizer, there's enough oxymorphone around to probably make that a different drug. Most of us don't make lots of oxymorphone. But certainly codeine to morphine, if you're getting any analgesia at all, you might be getting some analgesia. If you're a 2D6 poor metabolizer, you're not making morphine, or hardly any. But you might be getting some codeine 6-glucuronide. At least in the animal models, it's analgesic. So possibly. She was also on Tramacet. Well, Tramacet, it's an SNRI with the M1 metabolite, has affinity for the mu receptor in the nanomolar range, but the parent compound only has affinity in the micromolar range. And we tend to not get micromolar concentrations of that. So there might have been sort of a pattern here, but the problem that was overarching everything was she a CYP2D6 poor metabolizer? Was she a mu receptor polymorph like an A1 or an G? Or was it simply that she wasn't taking it enough? Or was it oral bioavailability? Or was it poor adherence? So what we ended up doing, she was still R1. She wanted to be cured if she could be. She had nociceptive somatic pain with neuropathic features, and we really did agree that she'd never had an adequate trial of opioids. So we started her and discontinued everything, got rid of the opioids and the Tramacet. You can see our doc put up 7.5 milligrams sub-Q Q4 and kept the breakthrough at the same. We're trying to see where she comes out. We switched her to gabapentin, made suspension. We start low, 100 milligrams at bedtime, and then move up from there. Also decreased her mirtazapine because we think for the first time she might have actually been getting it. What happened? So the gabapentin was slowly increased from 100 at bedtime to 300 TID, and she complained bitterly of sedation. She had ataxia, she had dizziness and vertigo to the point she got up one day, she fell, and when she fell, she started bleeding again. So it was terribly traumatic, so we titrated her back off the gabapentin. We changed her from hydromorphone to methadone, thinking that with the NMDA effects, that possibly that might be better. Well, she hated it. She was very dysphoric, she didn't like it, but it had been helping her nociceptive pain. Her pain scores really went, when she was on opioids, really shot up. So we put her back on hydromorphone. Her anxiety increased, her bleeding increased, and a good friend came from home and said, why don't you try some marijuana? And I thought, well, if you look at the Canadian Pain Society evidence-based guidelines from 2014, cannabinoids have moved from fourth line to third line. We've already pushed, we had a reasonable trial of gabapentinoids. She's been on TCAs back in the 90s and didn't do well on them. We're not sure if we should put her on an SNRI again. She had tramadol, but she had opioids, so we decided that we will, I thought it was a reasonable try. Now the doctor that was on that week absolutely hates and abhors, especially plant product marijuana. So we really had to push, and Gary Frank, the nurse consultant, and myself really pushed on her behalf to try to at least try this because it seemed reasonable. There was no way he was going to go for a plant product like Sativex. So where did we end up? We ended up with Nabilone. So what happened with Nabilone? Again, this was very, very careful with her feedback titration. We got her hydromorphine. She wanted 5 times daily. She was trying to take that larger dose at bedtime and not be awakened. And she liked to be more awake during the day and then at bedtime. So it was 4, 4, 8, 8, and 12. And I know that's a bit weird. She had more radiation therapy, which caused a huge flare in her neuropathic pain. So we started her on a tiny little dose of Nabilone because the doctor was really worried about it. Slowly titrated her up to 0.1 milligram BID. And she said, my pain feels better. She says, my anxiety feels better. And the other thing was that she said her pain scores were about 5 to 6 out of 10, which was her goal. That for her, she felt was fine. And she actually stayed on that, and she remained on that until her death. We tried desperately to get her home numerous times, and she did manage to get some passes, but she actually, her trajectory changed very, very quickly. But I learned an awful lot from this lady. I spent a lot of time with her as well, and she's just absolutely wonderful. So what I'm basically saying is, under these conditions, I have no problem with trying an opioid even on a lower level of evidence. But I thought we might need a soothing image interlude after that. This is Aaron. We just adopted him. He was racing all the way up into July. The other thing I want to say is I believe that there are puppy dogs in heaven. I don't know that there are puppy dogs in heaven, but how could it be heaven without puppy dogs, right? He's so earnest. I just love this guy. He's also a small horse. He's really big. So, yeah, okay, I'll hurry. So the other case we had, and this one really kind of surprised me because I came up thinking it was going to be for symptom control. He's 70 years old. He's widowed. He's got 1 daughter, 1 son. His daughter works in the hospital. She's got a clinical science background. I walk in. He's stage 4 metastatic colorectal. It was adenocarcinoma. He failed. It was either FOLFOX or FOLFIRI, I don't remember. And they were offering him some palliative chemo with irinotecan. And he said, I want some cannabis oil. I'm thinking, oh, he wants that to treat some of his symptoms. No, he wants cannabis oil to cure his cancer. He said, that would be the knockout punch. I've read all about it. And his daughter is looking at me like, no way in hell are you going to recommend that he have this stuff, right? And I'm like, well, and again, it's up to the patient, right? So, so I needed to hear him out. So I talked to him at great length. But then he kind of admits to me, oh, I actually have some. Yeah. Friend of a friend of a friend of a friend of mine, no names, right? He said, and I'd really like to try it. Well, the other thing is we'd had a lot of trouble with this guy. He had significant sensitivity to opioids. He had actually come in an active delirium. We were on haloperidol. So what was going to happen if he took some of this? And we didn't know exactly what he had. His oncologist said, no way, I don't want anything to do with this. So in the end, I actually ended up sending papers to the oncologist. Now we say the drug-drug interaction potential is low. At the same time, there's not really good studies. So the best paper that's out there is actually an in vitro paper. So anyhow, the doc agreed, I guess the risk was low. So, but we said, why don't you try this? And we'll see how you react to it. But the other thing is, we weren't sure exactly what he had. So I went to the authorized licensed dealers, find out what he bought, and lo and behold, he got it from Aurora. So you know that it's local. It's the Aurora Borealis. And what does it say? The healing power of nature. Nature would never hurt you. So you just sign up. This is and then you get a welcome package from the doctor, and the doctor registers the patient, and you click, 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 and you've got what you want. So we actually figured out that what he actually got did have higher. So they will give you THC and CBD levels, right? And he did have higher CBD levels, which is where the argument that it might work is. So we actually had him take one in the hospital, unbeknownst to some people. And the fact is he seemed to tolerate it reasonably well. He actually was discharged home, and he really didn't have a lot of symptoms. He actually ended up having 3 cycles. His daughter told me, he said he felt that it was helping. But his ECOG status continued to deteriorate. The chemo was stopped, and he passed away at home. So we need another soothing image after that. So 36 degrees, it was hot. So, but I want to just finish off. I know we're right at the end here. I want you to charge your flux capacitors with 1.2 gigawatts, right? So we can go into the future. And what I want to tell you is, so maybe the evidence isn't there yet, but there are a ton of studies registered, and it's not just with clinicaltrials.gov. There's some in Canada, there's some in Europe. So this one's the effect of cannabis on pancreatic cancer. There's another one, medical marijuana and pediatric CNS tumor population. There's a lot of studies that are coming. There's also a whole plethora of CB1, CB2 agonists, antagonists, cannabidiol agonists, and these vanillinoids, I didn't go into in great detail, but fatty acid amide hydrolase inhibitors, they break down endogenous cannabinoids. There's even a theory out there that fibromyalgia may be due to the fact that you just don't have enough endogenous cannabinoids, because it has been shown. They're already looking at single nucleotide polymorphisms of that enzyme in breast cancer. So there's a lot of research on it. So I think we might see something. The other thing would be better or standardized plants. And how many saw this on CBC? Yeah, it was really interesting. So they went to 7 Toronto dispensaries and got 12 popular strains. The mean THC levels were 20%. With some strains going as high as 30%, but there was no CBD in the samples at all. So we really do believe that there may be a difference in that. So they actually talked to Jonathan Page, adjunct professor at botany, University of British Columbia, who said, these guys are smoking 2 to 8%. Turns out Jonathan's got a company. The Science of Better Cannabis. Okay, and what they'll measure everything you want to know. So at the very least, they're trying to do some sort of standardization. They also have geneticists in their company trying to breed, not just breed, but genetically alter the marijuana genome to make more useful compounds. And where I want to finish off is this whole thing about exposure. How many have heard of dabbing? Dabbing has been called, yeah, it's the crack of pot, right? And so they basically extract from THC and butane oil. So they're getting, if there's any insecticides, they're getting that too, right? And they make different compounds. The purest of this is supposed to be shatter, and shatter looks like that because it breaks. And of course, the THC content in that can be 60% and up. So it's very high. And so the other thing is there's this quest for pure THC, this Gorilla Glue number 4. I'm going to take that for sure. The strain tested out at 96.75%. I'm not sure what you want to do with that kind of pure THC. They advertise what it's great about it is you get a pure unadulterated high, and it lasts a long, long time. Okay, you're just taking a huge dose of THC is what you're doing. The other thing is that has led to, and we've seen 3 of these in the last year in palliative care, cannabinoid hyperemesis syndrome. And it seems paradoxical. Well, it is paradoxical because people are smoking this stuff sometimes for nausea and vomiting. Then they start having more and more emesis, so they smoke more. The fact is, the way you identify it, it's always in patients that have been on it for a long time, but they take really, really hot showers because the hot showers, again, don't really know the mechanism, actually decreases the nausea and vomiting. There's even been people show up in emergencies, not here, but have scalded themselves because the nausea and vomiting is so bad, they take a shower to relieve it. And so there's this whole syndrome. This was a paper of 98 patients, Mayo Clinic proceedings, must be good, right? And the last thing I wanted to end off with spice and K2. Now these are synthetic cannabinoids. Now some of them actually could probably have clinical utility, but the fact is these things are so very, very potent that for the very first time, we certainly are seeing hyperemesis syndrome, seeing a whole host of neurological side effects. But what I wanted to end up with is, for the first time ever, we're seeing deaths. So I liken it to this. It reminds me of, so when you were in the jungle and you had some cocoa leaves and the locals would chew on them, it was like drinking coffee. Not a big problem. And that's probably what marijuana Cheech and Chong stuff was like. But when you're getting to things like this, now what you've got is it's not just chewing on the leaves anymore. You're extracting the cocaine. You're making crack cocaine. And now we're basically making crack pot. Sort of a joke. So my clinical conclusions are, I have no problem with careful N of 1 use of marijuana as plant products. I think that I even know one physician who does go online He selects different species that have, and he looks at THC CBD levels. And he claims he's getting pretty good results. He feels that he's helping people and he hasn't harmed them. And I think also with the, these variant types of seizures, these rare conditions, I have no problem with that. I think we can also use the synthetic compounds as indicated. I think we need to, that there will be better standardized products. And I also think we are really on the cusp of an awful lot more of information that's really going to tell us once and for all if we really can use this stuff as a drug. So hopefully by now, your belief system is shot. And I promised to give a whole bunch of references. So, and I did change the slides repeatedly. So if anybody wants the latest iteration, you can just email me at pmail@ualberta.ca, and I'll be happy to share that with you. And that's all I really wanted to say about that.